My talk is about reconstructing or trying to model past climate using records from small sediments of small lakes. And this is not meant to be an exhaustive, neutral sort of summary or review. It's going to be a highly personal, somewhat biased review or view of how I see challenges and also the joy of reconstructing or trying to reconstruct a paleoclimate using lake sediments. Okay, so why do we study small lakes? Small lakes have much shorter response time, so it's much closer to human time scale. Okay, archaeologists couldn't care less if climate changed on 1,000-year time scale. They want to know what happened in decade or even shorter time scales. So that's where records from small lakes often come in handy. And it relies on indirect or proxy records because instrumental climate record is quite short. So anything in sediment that responds to climate, if it's not altered, can be used. We always have the problem with equifinality, trying to distinguish between correlation and causation. Most of what we see, we have possible multiple causes, which we need to try to figure out, which is more likely than others. Okay, the kinds of lakes I've modeled mostly are in semi-arid locations. So most of my reconstructions have focused on dry wet cycles. People who are looking at, especially North America, at the most recent couple hundred years, maybe looking at land use changes. And in some other locations, it is possible to model when it's warm or cool. Okay, the many records are quite messy and it's not straightforward to interpret. And it's in big contrast to some of the well-known paleoclimate records, such as deep sea record, such as benthic stack, I'm showing Lisek Ramo 2004, or Antarctic ice core record, which shows glacier to glacier cycles, or some of the spirothem records. I'm showing one from Hai Chen, 2016 paper, and his co-authors. So East Asia monsoon regions. So it's the one dominant factor that affect precipitation amount in these regions. So the green curves you see in label D, that's a splice of multiple capes that are affected by East Asia monsoons, and they show good correlation with insolation curves. Okay, some of the really long-lived lakes, Lake Agugeten, this is a crater lake, has a record going back 2.8 million years. And I tried to stretch the record as much as I could horizontally. And you can see that the total organic carbon and diatom productivity are high during the high insolation periods. And in some periods are showing superinterglacials with mean temperature warm months reaching over 10 degrees in location in Siberia. That's quite amazing. Okay, now back to what factors affect lake sediment records. So tectonics and glacial isostatic adjustment, it can reverse the flow of water. If the lake is long, north-south, it can cause stream capture, piracy, earthquake-induced landslides. All those things can change the hydrology. Okay, but the big factor in small lakes is catchment geology and hydrogeology. So what kind of bedrocks do you have? Are they carbonates or not? Okay, because rainwater, which is the ultimate source of lake water, is primarily just water and carbon dioxide. So it's a very weak acid. Okay, as when rain falls on the ground surface and starts to percolate through soils, soil is very acidic because of all the respiration. So the water becomes even more acidic and more reactive. So it can do quite a bit of weathering and then once it enters below the water table, be a lot of weathering reactions, or you might call rock-water interaction, that brings in cations and anions from the rock that the water is flowing through. So it's no longer just water and carbon dioxide. Okay, climate also affects the lakes. Seasonality, is it mild temperature all the time, or is it continental with hot summers and cold winters? Does it freeze in winter? So it affects the overturn of the water column. Is it hydrologically open or closed? Okay, if in very wet places, you don't see many lakes because you just see streams. It's hydrologically open. Whereas in semi-arid locations, we see a lot of hydrologically closed lakes. Okay, so whether it's open or closed, that affects hydrologic budget, residence time, which in turn affects response time and how intensity changes are recorded. And of course, water chemistry in hydrologically closed lakes, water evaporates, leaving solutes behind, which makes the water more brackish and saline. Okay, and types of climate make a difference. And biology also affects sediment records. What kind of vegetation is growing in the catchment basins? Okay, for instance, conifers are well known for turning soil acidic, so that more acidic water comes into the lake basins. Okay, biota in water itself. Okay, algae will induce carbonate precipitation when its photosynthesis is very active. Biogenetic carbonates, okay, ostracods for one, snails, okay, they take dissolved calcium and bicarbonate out of water to make calcite aragonite. Biogenic silica from algae such as diatoms. And if we have sulfate reducers, we make sulfides, and that's left in a sediment record. Okay, humans also affect the lake sediment. Especially in North America, when Euro-Americans came in, they cut down the trees, so they turned over prairie and increased the erosion, and then increased the eutrophication of the lakes. If they build dams or levees, it also changes hydrology. Okay? And one of the big problems is anthropogenic effects on climate hindcasting. Okay? We often try to calibrate using instrumental records. Okay, instrument records start when human effects become really important in North America. Okay, so what we're using for calibration may not be all that good if you think about what sedimentation may have been like before people came in and mucked up the landscape. Okay, so what do we use as proxy records? This is not an exhaustive list. Pollen, so we use wind-pollinated catchment vegetation. Insect-pollinated pollen are usually too heavy. They don't get carried by wind, so it's wind-pollinated vegetation. Diatoms, okay, so they're used to reconstruct salinity or nutrient levels. Ostracods, okay, so diatoms are plants, ostracods are animals, often used to reconstruct salinity or carbonate alkalinity to calcium ratio. So it's one of the few proxies that can actually tell you something with the water chemistry in addition to just concentration of ions. Chironomids have often been used as temperatures. These are non-biting midges. And of course, a whole bunch of stable radiogenic isotopes or carbonate minerals, ostracods, whole bunch of things. You can try to say something with the water or carbon cycle, something about temperature, source of detritus, etc. Whole bunch of applications and possibilities. Element ratios, okay, something about salinity, water chemistry. Okay, we need something that record element ratios. Okay, and we tend to forget to look at the matrix itself, but the grain size, mineralogy, or composition of matrix tell us a lot about strength of the flow. River water has an inflow, a source of detritus, what kind of deposition environment was present. And some people use magnetic properties so that they can say something with the pedogenesis or erosion that bring magnetic minerals into the lake water, into the lake sediments. Okay, just a few examples of biological proxies. Upper left is pine pollen. So the scale, lower left two are centric and pinnate diatoms. Okay, diatoms go across 10 microns. And then the right-hand side are ostracods. Up diagrams are dead ones. And then lower right is live ostracods, which actually have patterns and whatnot. All the pigments get destroyed after they die. Okay, ostracods that look like they have parts, it's dead version. Okay, so how do we use proxy records? Uniformitarian principle is used. Transfer function is a very popular method. We make sets that relate to faunal, floral, some parameter. Okay, so we might collect a whole bunch of items from many lakes in general region, along with environmental information. Okay, so you might collect water temperature, etc. Okay. Or modern analog technique, we relate fossil assemblage to modern assemblage, often used with vegetation, but with other things as well. Okay, and in both of these techniques, there's an assumption being made that we already have an idea what is the important environmental factor that is affecting distribution or presence absence of different species, say of ostracods or diatoms. If you don't know what they are and are not measuring it, then no matter what kind of ordination you conduct, you're not going to find what is affecting the distribution of the fauna or flora that you're studying. Okay, the third one that I rely on quite a bit is oral ecology of individual species. Okay, so it's complete life cycle, when they molt, when they hatch, where they bloom, and how they react to different environmental factors. Okay, so here's an example, transfer function by Stefan Mischke and co-authors. So it's Levant. 178 sites were sampled and 123 had full environmental data and ostracods they were kept. And then from that, found 42 ostracods, and of that, only 30 occurred in 3 or more samples. Okay, so we don't want to end up using something that only exists in one location, which may have been brought in by, say, aquatic bird, and it's not really native to that lake, so those are eliminated. Okay. So on the right-hand side, you can see 30 species with their range of specific conductivity. Okay. And so after the ordination, specific conductivity was found to be the dominant factor in distribution of these species. Okay. And then a weighted average partially squared method was used, and then a middle figure shows how well the training set works against the actual measured specific conductivity. Okay, in modern nano technique, this one was used to reconstruct the temporal changes in prairie woodland ecotone. So on figure A, the brown line is the division between areas to the northeast, which has many more trees, and then to the southwest, which is more prairie. Figure B is from the remote sensing data. Figure C is what the modern vegetation looks like. Okay. But you verify that modern pollen assemblage looks like the actual modern vegetation, and that there's a good correlation with temperature and precipitation. Okay, then with that, you can take sediment core from some area within this region and then start looking at the pollen assemblage and compare against modern analog. Okay, so you might find that 6,000 years ago, the pollen assemblage is similar to what exists in some area to the south of the current location. Okay, and the use of water ecology, going on the left side, water chemistry ranges for 5 ostracod species. So you notice that on the x-axis is total dissolved solids in milligrams per liter, and on the y-axis is the natural log of carbonate alkalinity plus calcium in milliequivalent ratios. So looking at the charge against charge. And you see that some species prefer to be in or found in waters that have low alkalinity to calcium ratio, and some are in high alkalinity to calcium ratio. And in dilute waters, they're all staying sort of near alkalinity to calcium ratio of 1. Okay, and then USGS has the fantastic record looking at temperature precipitation range for a whole bunch of trees, both deciduous and then for white oak. So our left shows today in of January, July temperatures, January precipitation, etc. So the faint gray areas are all the climate combinations that are found in North America, okay, and then black parts indicate where white oak is found today. Okay, going back to the equifinality problem, which is not just for small lakes, but I think it's particularly dire for small lakes. So existing no handcasting construction because nobody was there when changes were taking place. We're looking at finished experiments and trying to figure out, okay, what happened? Okay, and small lakes, they're much more influenced by what's going on in the catchment. So compared to marine sediments, so polar ice sheets and monsoons, equifinality is a much more serious problem. Okay, so we can tell some changes occurred, but what caused the change, that's a whole nother problem, challenge. So how do you examine possible causes? Okay, one of my favorite, if you can, is lake to extent, is to come up with monitoring studies. Temporal is what I've done. It was training sets and analog studies. They're looking at spatial variations, so they're sort of substituting space for time. So try to understand the modern system as much as possible. In nearby lakes, you might look at pair of lakes from same climate and geology, but with different hydrology to see how they might react differently, and then use multiple proxies. Understand each proxy, which detail do you have to understand for each proxy, and what we find is observed change, necessarily synchronous or of similar magnitude. So plane response on a synchronous similar magnitude. So example here is ostracods. So it shows 4 possible scenarios for changes in ostracod fauna assemblage as in response to changes in shell chemistry. Okay, here assumption is that shell chemistry is related to water chemistry and with delta C13, a carbon cycle. Okay, so assumption being made here is that climate change caused shell chemistry change, which may or may not be true. Okay, so take home message I like to take you away is no matter the size of the lake. Okay, there are 3 things that are going on: catchment processes on landscape, within the groundwater system, which I think is it's my favorite, confounding factor, groundwater, and the process within the lake. Okay, so effect on small lakes from the first 2 is very large, so proceed with caution when you're trying to reconstruct or model past climate or environment using sediment form small lakes. Okay, so I'm going to use 3 examples. Hope I can have time to finish them. So first, I showed nearby lakes with different hydrology, or actually permeability in the aquifers. So 2 lakes from Montana. Second one is example multiple proxies, the Elk Lake in Clearwater County. And the final one is a different type of multiple proxies, which is coupled hydrogeologic field work and groundwater flow simulation, also from Elk Lake, but it's from Grant County, Minnesota. Okay, every county in Minnesota has at least 1 or 2 Elk Lakes, in addition to Cedar Lake, Lone Lake, etc. So I need to specify which county they're in. Okay, so these are the 3 lakes. P minus ET, precipitation minus evapotranspiration is usually positive. Elk Lake, Grant counties, P minus ET about 0. Evans and Jones Lakes, P minus ET is negative. Okay, I'll start with the Evans and Jones Lakes. Okay, so the lakes are 6 kilometers apart, have the same climate. Geology is similar, although Jones is located with a lot of glacial outwash and alluvium, whereas Evans is located wholly in the glacial till, which is much lighter, with much lower permeability. And you can see that Jones Lake has reversed that. Jones is 1.5 years, and this is 2 years residence time. Okay, so they deviate from church global waterline. Okay, this here is showing much farther evolution from the precipitation that fall in the area. Okay, this lake has solids between 180 and 280 millimoles per liter, and calcium ratio, which is quite high because all the calcium that comes in through groundwater is immediately precipitated out as aragonite, so that lake water has no calcium left. And it's also use sulfate enrichment, but when you look at carbon alkalinity of calcium ratio, it's still increasing because calcium removal is so complete. In Jones Lake, with a shorter residence time and much higher loss of lake water groundwater system, TDS remains quite dilute, and the calcium ratio also remains quite low. Okay, and what you see is that the Jones Lake, because of its water chemistry, has types of mustards that are in this green zone, dilute waters, whereas Evans Lake has substance which is red zone. And so if you look at statistics without having to soil work that Mark Shelby has done, you get the feeling that these 2 lakes are in a different climatic regime. Okay, and but both lakes show consistent pattern. Okay? It shows the effect of human settlement, increase in erosion. Okay, so that relative proportion of carbonate or orthogenic minerals decreases. Okay, so the sense of timing, effect of human settlement are recorded in both, but Evans Lake makes you think it's in the arid environment, and Jones Lake makes you think it's in the humid environment. Okay, so hydraulics of the aquifers really affect how lakes behave and what kind of sediment is formed. Okay, let's move on to Elk Lake, Clearwater County. So right about here, so it's in the P minus ET positive. Okay, and it was studied by a whole bunch of people from USGS and from University of Minnesota. all came out in special paper, JSA special paper in 276, 1993. It's all black and white. So here's Elk Lake. Oops. Sorry, I just lost the screen. I can't see what I'm doing. Okay, so Elk is the source of the Mississippi River. So it's located on the edge of the low-range mixed forest, conifer forest. Okay, it's a deep lake and it's permanently stratified at the bottom. It's barbed sediment. So the chronology is we're counting varves, annual couplets between precipitation. Okay. And the vegetation is typical of what we see in northern Great Plains, stag holes taken over by warm dry climate, oak and grass. There's more, and then be better birch, and then back here when it's drier from the spruce forest, we're looking at the jack pine. Okay, so vegetation reconstruction tells you that it went from the early Holocene cold climate with spruce trees, changing to jack pine, and then prairie, which is warmer and drier, and coming back to slightly cooler and wetter climate. Okay, if we look at things that were living in water, okay, first of all, look at diatoms, benthic diatoms. Okay, diatoms are photosynthetic algae, so benthic diatoms, you're not going to find them unless enough sunlight reaches. Okay, so the benthic diatoms stay relatively low in concentration, less than 20%. Okay, so the environment didn't change all that much. Okay, and the ratio of Stephanodiscus minutus and Fragillaria crotonensis, that tells you the difference between overturn, which is preferred by Stephanodiscus, which requires a lot of phosphorus. So when water column overturns, bringing nutrient from the bottom, Stephanodiscus goes crazy. Okay, whereas Fragillaria crotonensis and Olaxera prefer waters with higher silicon and then lower phosphorus. So stormy summers and these summers that bring a lot of aeolian sediment that favors Olaxera and Fragillaria. Okay, so during the prairie period, there are fewer trees, so there are less shielding of the lake from the wind effect, so there's much more mixing. Okay. And from Ostergaard's fauna assemblage, modeling says that during the prairie period, the dissolved soil is a little bit higher. Okay, but if you look at delta O18 of endogenic carbonates in Ostergaard's, there's just a little bit of change toward higher values, but not much. Okay, so it's pretty remarkable. We look at the replication for indicator of water it's and even that time, even though there are relative changes in abundance of different species, there's no wholesale for into a different type of environment. Okay, so Elk Lake is an area that's where the ecotones from the mixed forest to deciduous forest of prairies place really closely together. So it should be sensitive to climate change. And yes, vegetation change before that. But because of the really large groundwater inflow, water chemistry doesn't change that much. Okay, so what you see is the effect of the loss of forest that shelter Elk Lake from the wind. But overall, there's very subtle effect on diatom assemblage and not a huge effect on oxygenized observed carbon, dissolved carbon. Okay, so last example. Sorry, it's taking too long. Grand County. This is an area that is close to prairie, parkland biome, and not too far from geospheric biome. It's a glaciated landscape with a lot of chill, with some fine sand features. And like here have been for sample under the study. We have groundwater from 2 different locations, streams, Lake, right here. There are 2 types of groundwater that's discharging. One dominated by carbonate, the other is dominated by sulfate and bicarbonate. Okay, so 60 meters sediment, and overall climate change is expected very similar to Elk Clearwater and Northern Great Plains in general. All across species abundance, so cool to warm, maybe warm freshwater species are found in zones A and D. Species found in zones B, B2 tolerate hydro dissolved solids, and in zone D, warm, cool, freshwater species. Okay, we have isotopes, magnesium calcium, strontium calcium ratios from ostracod shells, single species, and then reconstructed bicarbonated calcium or carbonated alkylated calcium, mean annual precipitation, total dissolved solid. Those were reconstructed, modeled from ostracod fauna assemblage. Okay, so B to B2, generally more concentrated water, less precipitation, okay, and then bicarbonate calcium ratio is higher in the zone D. Okay, and zone B2 was rather confusing at first, as this is the peak of the prairie dry period. We expected delta 18 to be consistently high, but either we have low values. 2 species, Plenary and Heterotensis, come to rescue when we need to figure out what's going on. Heterotensis live near one water seepage areas. So it indicates that the core location, which is from the deep heart of the lake, had so little water that the groundwater table was at that level. Okay, and Luminosis species that tend to be in bicarbonate depleted water, so it suggests that there's a lot of sulfate. Okay, so the deeper aquifer that is sulfate rich was discharging into lake at that time. Okay, so Holocene, cold and humid, freshwater. So and that period is more to mid drought and sustained drought. Shows depression when you the spring lake shower can put ice topically and then totally into a lake, whereas during the peak of the spring drought, all the static sediment precipitation, the area comes straight into Elk Lake. Okay, and we really needed field work, flow modeling to make sense of the data we got from zone 2. Okay, so in summary, getting just on site, like the Jones and Evans lakes could have resulted in a model that's not easily relatable to modern climate. Okay, and that would work if you have pairs of lakes with different types of sediment record. Even if the lake is now dried out, we can still pull out those information. Okay, monitoring modern system processes. I think it's really important. I always try to do that whenever possible. and then also rely on modern multiple proxies. And it's not popular these days, but I'm particularly fond of pollen data because it tells you about what's going on the landscape. Okay, thank you very much.